So, good evening, ladies and gentlemen, and welcome to the Royal Society. My name is Adrian Sutton. I'm standing in for the chairman of the public engagement, introduce Dr. Nick Lane. But before I get on to that, housekeeping points. First of all, please turn off your mobile phones. Putting them on silent is enough. But we really don't want to hear any ringtones of any kind. There are no planned fire evacuations, so if you hear the fire alarm, it's serious. The fire escapes are those doors and also the doors in the middle on your right, over there. The event is being webcast and it's going to be recorded for the Royal Society's archives. And we will also be using a speech-to-text service for people who are hard of hearing. So tonight's talk, entitled "Why is Life the Way It Is being given by Dr. Nick Lane, winner of the 2016 Royal Society Michael Faraday Prize. Let me tell you a bit about that prize. It's awarded annually for excellence in science public engagement. It recognizes a scientist or engineer whose expertise in communicating scientific ideas in lay terms is exemplary. Dr. Lane can count himself amongst a list of exceptional science communicators, including Frank Close, Jim Al-Khalili, David Attenborough, Francis Balkwell, Jocelyn Bell Burnell, Marcus de Sotoy, Brian Cox, Andrea Sella, and Catherine Willis. Dr. Nick Lane is a reader in evolutionary biochemistry and biochemist in the Department of Genetics, Evolution and Environment at University College London. His research is about how energy shapes evolution, in particular the way in which the very peculiar mechanisms of biological energy generation forces evolution down unexpected paths. This perspective has tremendous breadth, cutting right across the whole history of life, from its very origin nearly 4 billion years ago to the singular appearance of all complex life on Earth, to the structure of our own cells and the way that affects our lives and deaths. Nick leads the UCL Research Frontiers Origin of Life program and was a founding member of the UCL Consortium for Mitochondrial Research. His work has been honored by several awards and prizes. But he is best known for his books on the evolution of life, which have been translated into no less than 25 languages. His 4 books treat the grand sweep of evolution, from the origins of life to our own aging and death. His books have been critically acclaimed, being named among the books of the year by Nature, New Scientist, The Economist, The Independent, The Times, The Sunday Times, The Telegraph, and The Wall Street Journal. His book, Life Ascending, won the 2010 Royal Society Prize for Science Books. He was described by The Independent as one of the most exciting science writers of our time, and his most recent book, and I happen to have a copy of it, The Vital Question, even came to the attention of Bill Gates, who described it on his blog as an amazing inquiry into the origins of life. Ladies and gentlemen, I'm pleased to present Dr. Nick Lane. Good luck.
Thank you very much for the very kind introduction, and thank you all for coming. I'm astonished that so many people are here, and I'm told there's an overflow room which is also full, so thank you very much, everyone, for coming. It's a huge honour, I hardly need to say. The list of winners of this prize in the past are really number many of the people who inspired me as a child and as an adult, and to be on that list is a bit overwhelming. Anyway, I'll talk this evening about why is life the way it is. Now, I don't know what you make of that question. This is actually the subtitle of the book in English, and I try my subtitles out on people, and I called a friend and said, well, the subtitle will be, why is life the way it is? And she said, yeah, it's awful, isn't it? I need a holiday. You never can quite predict how people are going to react. I had probably the best title and subtitle of any of my books was called Power, Sex, Suicide, Mitochondria and the Meaning of Life. And I had to find some way, really, of trying to sex up mitochondria, try to get people to read a book about mitochondria. And it's strange because I've noticed it's not cited as widely as I would wish or as other books are. And I think people are a little bit ashamed of citing a book with such a salacious title. It looks as if they've not been spending their time wisely. And it's also curious that people can only remember one word from that title. And the best review I ever had said, I gave up smoking after reading Nick Lane's first book about oxygen. So I approached his book on sex with some trepidation. Anyway, this question for tonight, why is life the way it is? Even in a scientific context, it might seem a strange question because you might think that life is just about any which way. And these are just animals, and I'm told the blobfish is real there. But it seems as if just about any morphological form or shape or way of life or just about any way of being exists on the Earth. And so, how can that question make any kind of sense? If we look at a tree, a genetic tree which shows the relationship between different animals, we see something like this. We are here, you might just be able to read that. It looks again as if all the genetic space has been explored thoroughly by the animals that exist already. And we can go much broader than that, of course. This is the entire tree. And so we include plants up here, animals are over there, protists, so single-celled organisms, fungi down here, bacteria and archaea. But I need to stress that this is not a tree of life. It makes me think of Magritte, who said this is not a pipe, which must irritate some people. I read this quote that he says, well, can you stuff this pipe? No, of course not. It's merely a representation of a pipe. And what we need to understand about the tree of life, of course, is that this is just a representation. But it goes beyond the problem with the representation. The representation, it looks as if all of this space is completely full, as if there's almost nothing left to explore. And that's an issue with the presentation of it. But it's also a tree of a single gene, I beg your pardon, a single gene. And if you were to construct a similar tree from a different gene, you would get a different tree. The branches would be in different places. And so it's a tree of a gene, and if you try and do a tree of life and you construct it from all the genes, you get a very different picture, and I'll come on to some of that. The other issue with this tree is that the bacteria and the archaea down here look trivial. It looks as if there aren't really any and that they're not very important. They're down at the bottom of the tree. It's a very misleading impression. And again, you can understand if what we're really interested in are the animals up here, then that's what we've put the focus on. But as a representation, it leaves something to be desired. This is going back now 20 years, and this is an equivalent tree using the same gene, and it has a lot more perspective. And it raises some really interesting questions. So this is what's known as a three domains tree of life. And it was discovered by Carl Woese, who looks rather like James Dean in Rebel Without a Cause. And he pointed this out in 1978, discovered this group that look a lot like bacteria called the archaea. Nobody really had much of an inkling that they existed at the time. And he came up with a tree, this was 1990, showing tremendous variation within the archaea. The lengths of these branches give an indication of the amount of genetic variation within each group. And so the bacteria are over here, the archaea here, and then animals, plants, fungi were constrained to this small corner of the tree. It's another Copernican kind of revolution which pushes humanity across into an uncomfortably small corner where we're marginalized from importance and it feels uncomfortable. But as soon as you look at this tree, it begs two questions. And this is one theme that I'd like to try and bring out from this evening's lecture, that science is not really a collection of dusty facts at all. It's a way of seeing the world. It's a way of testing those questions and trying to understand how the world might work. And the kind of questions that we can ask about this are very simple. They're almost childlike questions, but it is easy to miss them. So why is it that these two groups have got so much genetic variation and yet remain so simple? In terms of their morphology, they really haven't changed very much. There's nothing, certainly nothing like Carl Woese built from bacterial cells. It simply doesn't exist. So what was happening down this branch of the tree of life that wasn't happening over here? And you can tell just from looking at that that it was not really anything specifically to do with genes, because there's plenty of genetic variation there. It just didn't lead to morphological complexity. So these are quite simple, childlike questions, but they hit you in the face as soon as you look at that. They don't hit you in the face at all from this. So again, it comes down to the representation. So there have been, what would happen, I suppose, the question here is what would happen if you were to wind back the clock? If you were to come all the way back to the origin of life and let the clock play forward again, would we end up with something like that or something quite different? And there's no agreement. This is another theme from science that I'd like to get across, is that there can be tremendous intellectual arguments about the meaning of things. And this is a very good example. So on the left-hand side, as you're looking at it, we have Jacques Monod and Stephen Jay Gould. So Jacques Monod wrote a wonderful book called Chance and Necessity, right at the dawn of the molecular biology era. And it was a fairly bleak outlook. He really thought that life was almost impossible to get going and that we were alone in an empty universe, and it was really a French existentialist philosopher's view of life. Stephen Jay Gould wound back the clock in Wonderful Life to back before the Cambrian explosion, when the first animals appeared in the fossil record. And then let it play forward again in his imagination and said, what would we get? Would we get human beings? Would we get vertebrates? What kind of animals would we actually get? And his view, and it was simply a view, is that you would get something very different to what we have, that the evolution that has taken place is effectively contingent on the circumstances, that you have a meteorite that wipes out the dinosaurs, and so that gives the mammals a chance to get a foothold, and so now we have mammals, but it could have been very different. On the other side, we have Christian de Duve, another Nobel laureate, as is Jacques Monod, and Simon Conway Morris, who was actually one of the subjects of Stephen Jay Gould's book, and they both think far more in terms of convergent evolution, the structural importance, that if you want to fly, you need to have something like wings, otherwise you'll never get airborne. And so there are engineering principles that force life down particular avenues, and they see those avenues as being the most important. And so these are different perceptions, but it's important because you can say that it's simply counterfactualism, but the question is, can we predict anything about why life ended up this way rather than that way? Can we imagine what life might look like elsewhere in the universe? What kind of principles do we have that will allow us to get at those questions? And so these books written by these four are a wonderful way of exploring the problem and laying out hypotheses. But the key thing from a scientific point of view is most of those hypotheses are testable in one way or another. It depends on your approach to the question. Well, here's how I see it. This is an indication of what's happened to bacteria over 4 billion years, and they've been flatlining. They ended up with, about the same degree of morphological complexity. We can see bacteria and archaea in the fossil record from 3.5 billion years ago, and they look a lot like that. They look a lot like modern groups that we can see today. So what have they been doing? In their biochemistry, in their molecular machines, they are fantastic. But in terms of their morphological complexity, they're surprisingly limited. So what was going on? And why is it then that complex life, everything really that we can see, has a common ancestor? It appeared once after about 2 billion years of evolution. And all of this amazing array of different life forms are all very closely related and share a common ancestor which arose around then. So this is another way of looking, if you like, at the tree of life. It's a very different one. Something abrupt happened, something odd happened, and we don't know what it was. I say we don't know. That doesn't mean to say we don't have ideas. We have plenty of ideas. The question is, how do we know which one is correct? So what forces constrain the evolution of bacteria? Why is it that they didn't become large and complex? Why don't we have bacterial humans? How was it that the complex cells escaped, the eukaryotic cells? I'll talk more about them. And would any of these forces be similar on other planets? Can we make a guess as to what aliens might actually look like? What kind of constraints from first principles would apply to them? So if we're going to ask this question, it's really the question, what is life? How do we think about it? And this is Erwin Schrödinger, who wrote a famous book on that theme in 1943, I think. And there were two famous ideas that emerged from this book. One of them was the idea that genes are a code script. And that was the first time anybody had used the word code script, or really thought in terms of information in biology. And he was absolutely correct. This was before DNA had been discovered. He was a direct inspiration to Watson and Crick and many others. The second theme of the book, though, was how life maintains its organization over time. Why don't we just fall to pieces, as entropy would tend to suggest we should do? And he said, he used rather odd terms, he talked about life feeding on negative entropy, or neg-entropy, he called it. And he talked about continually sucking orderliness from its environment. Now, these are slightly odd terms to today's audiences, but it's a wonderful book. He also had in a footnote, he said, if I'd been catering for physicists alone, I would have let the discussion turn on free energy instead. And I would say, to put all of that into more modern terms, he's saying something like life is the harnessing of chemical energy in such a way that the energy harnessing device makes a copy of itself. That's certainly how I see it. And he's linking the two key themes of biology, which is information and energy, together. So how do we generate energy, for want of a better phrase? Well, these are our mitochondria, the powerhouses of cells. So these are membranes inside, and this is where respiration is working at the level of oxygen and food reacting together to produce the energy. So there are, in all of our cells, we have in the order of several thousand mitochondria, hundreds to thousands of mitochondria. And if you were to unravel all these Christie membranes here and lay them out, you'd have about 4 football pitches worth of membrane as a surface area where respiration is going on. And what's happening, and I'm not going to get into any technical details, but effectively what we're doing is we are stripping electrons from food as we're respiring. And we're passing them in. Sorry, I keep pressing the wrong button here. We're passing them in. This is the membrane here, so I'm talking about these membranes. And inside these are these giant molecular complexes. I'm just symbolizing them as balls. So we're stripping electrons from food, and we're passing them down this is called a respiratory chain to oxygen. So we have a current of electrons. It's really as simple as that. And that current of electrons is powering the extrusion of protons across the membrane. So we now have a reservoir of protons on this side. And here we have what's called the ATP synthase. And this is driving energy production. It's a kind of electrical turbine in the membrane. ATP is the energy currency that drives absolutely everything in our cells. Now, these blobs that I'm showing you, the level of understanding required to understand how these work, I just want to give an indication. So John Walker won the Nobel Prize in 1997 for the structure of the ATP synthase enzyme. This is the turbine in the membrane. And this is from Sir John's own webpage. This was the state of knowledge in 1994. By the year 2000, he knew a little bit more. At that point, there was no structural information on the main part in the membrane, only by today. So this is over 20, 30 years or so that this information has gradually been pieced together. And this is another key aspect of science, is the time it takes. It takes decades to really get at these, and it requires determination and drive on the part of individuals, and of course funding for those individuals over those kind of periods of time to go from a very partial understanding of how something works to an extraordinarily detailed understanding of how it works. We need to know that, but we also need to conceptualize what does it actually mean, and the easiest conceptualization for me is simply a hydroelectric dam. So this is very equivalent to what I've been showing you. The water is equivalent to the protons. The turbine in the dam itself is equivalent to the ATP synthase. This is how respiration works at the level of cells. Now, this idea was shocking. It goes back to 1961 to Peter Mitchell. Here's Peter Mitchell. This is in 1947 with Jennifer Moyle in Cambridge at the time. And he put this idea forward in 1961 that respiration worked by pumping protons across a membrane, and he called it chemiosmotic coupling, and it was very mathematical, his approach, and physical, and he tended to put a lot of people off. A lot of people got quite angry because they didn't understand quite what he was talking about, and he had a knack of winding people up. And so it took a long time for these ideas to catch on. There was what was known as the Oxford Wars, which was a period of particular acrimony where people shouted at each other at conferences and so on. And in the end, it turned out, another finding of science, that pretty much all of them were, in fact, correct in one way or another. But Mitchell himself was the person who conceptualized the entire thing. Mitchell thought the ATP synthase was not going to work in the way that it does as a rotary motor and physically, mechanically bind phosphate onto ADP. He swore that was not how it was going to work, but that is how it worked. Anyway, the overall idea, I think Leslie Orgel captured it very nicely. He said, not since Darwin has biology come up with an idea as counterintuitive as those of, say, Einstein, Heisenberg, or Schrödinger. It really is an unexpected idea. I think until then, people had thought in terms of chemistry, molecules interacting directly with each other, and the idea that there would be an intermediary, a structural intermediary, of a proton's difference across a membrane was shocking. Now, Mitchell himself obviously saw this big picture from the very beginning. This is from a conference in 1957 in Moscow, where just about everybody who was anybody at the time was there. So JBS Haldane was there, J.D. Bernal was there. In fact, all the communists, they were all there. But Mitchell gave a talk at that same meeting. So he said, I cannot consider the organism without its environment. From a formal point of view, the two may be regarded as equivalent phases between which dynamic contact is maintained by the membranes that separate and link them. So he's really dissolving the barrier between the environment, the outside world, and the inside of the cell. And it's a very novel way of seeing things. And I think this is important and has been largely overlooked. Mitchell himself, incidentally, that's 1946. Here's from around the time he won the Nobel Prize. And I'm very struck that his hairstyle hasn't changed at all in those 30 or 40 years. He looks exactly the same, just a bit older. So there is a big question about that, though, because this is a very complex way of structuring energy conservation. Why would you have a membrane? Why have this intermediary? Why should it work that way? And how could it possibly have evolved in the first place? Because it's inherently complex. But the fact is, it's used by all cells. It's as universally conserved across all of life as the genetic code itself. It's a shocking statement, really, that. But this is just used by pretty much everything. And so you would think that it goes right back to very close to the beginning. So how on earth could it have evolved in the first place? Well, we can get some clues from the cells that are the simplest kind of cells today. So methanogens, for example, they're archaea. And acetogens, these are bacteria. So the archaea, if you remember, is one of the three domains of life. The bacteria are the other one. These are the simplest of cells. But these are from opposite domains. And they both live from a fairly simple reaction between carbon dioxide and hydrogen gas. And they get all the energy and all the carbon that they need to grow from that reaction alone. They don't really need anything. They need nitrogen for making their proteins. But in terms of the requirements for growth, all the energy and all the carbon they need comes from this reaction alone. It's what Everett Schock called a free lunch that you paid to eat. But there's one thing I really want to bring your attention to here, which is that it's not easy to get hydrogen and CO2 to react. If we could do that, we could solve global warming because we could strip CO2 out of the atmosphere, and we could solve the energy security because we can produce synthetic gasoline. So there must be lots of people in secret labs around the world figuring out how this goes on, and they won't publish it. So I don't know. So I have to bring my own ideas to the problem. And, well, I am a biologist and I see what these cells are doing. And over the last 20 years or so, we now have a much better idea of what these cells are doing. And one thing that they need is a proton gradient across the membrane. They can't grow without that. And if you look at roughly what's going on, don't be intimidated by this. This is an energy map, if you like. If it goes uphill, you have to put energy in as a barrier. This does not want to happen spontaneously. If it's going downhill, it's going to happen spontaneously. So you start with CO2 here, you react it with hydrogen. The first couple of steps to get to, this is a formaldehyde here, it's uphill. You have to put in a lot of energy to get that to react. This is why it's not an easy thing to do. This is why it's not economically feasible at the moment to make synthetic gasoline from CO2 and hydrogen. So what do the bethanogens do? Well, they use a proton gradient across the membrane to lower that barrier. They reduce the barrier to it happening, and then it can happen very quickly. And they get all that energy from lowering that barrier with a proton gradient. Well, that points, to me, to a particular environment on Earth where life might have got going. And these are alkaline hydrothermal vents. And they were discovered in the year 2000, so relatively recently, by Deb Kelly, who was the captain of the Alvin submersible. Actually, they were discovered by a PhD student who was on the trip as well. And these are about 15, 20 kilometers off the axis of the mid-Atlantic ridge. So there's nothing interesting there. Everybody knows that. So they were just getting on, writing their emails or whatever they were doing. And the PhD student was the only one who was looking out of the window and said, "Hey, wow, what's that?" And it turned out to be an entirely novel hydrothermal system. Nobody had seen anything quite like this before. People had predicted that they might exist, but nobody had seen one like that. It's not like a black smoker. You don't see, black smoke welling out of the top of these things. In fact, it was called Lost City in part. It was on the Atlantis Massif, and so Atlantis was already linked to it, but also it looks almost abandoned. There's a strange empty feel to this vent system. So this really is, again, another theme of science. This is a serendipitous discovery. It's really exploration, and I can't overemphasize the importance of just exploration in science, finding out things about the world, looking at what there is to see. So these vents I mentioned had been predicted. They'd been predicted by this guy, Mike Russell, about 10 years earlier. And his ideas at the time were really considered a little bit left field, probably, for most people. In some respects, they still are. But the discovery of this vent system, which conforms so closely to the kind of properties that he said they should have, really made him famous overnight. This was an article by Nature, the leading science magazine, and they dressed him up as Erasmus, and they called him Naissance Man, as in the birth of life, and linked him to the Renaissance and dressed him up, I think, as Erasmus. But the one thing I noticed from that is how much sartorial standards have declined among scientists over the last 400 years. I'm doing my best tonight to try to stop the rot, but I don't wear a tie often. So what Mike Russell argued is that these vents, if you look inside them, there isn't a central chimney. You just have this kind of porous rock full of, it's like a sponge, a mineralized sponge. And the hydrothermal fluids percolate their way through this sponge. And so you have high amounts of hydrogen and carbon dioxide, and you have natural proton gradients. You have these natural proton gradients because the hydrothermal fluids are very alkaline, and the oceans 4 billion years ago certainly were acid. Today they're mildly alkaline, but still far less alkaline than the fluids. And so he said, this is very equivalent to cells as we know them. This is what they look like if you go down to the level of inside one. So this is perhaps a few micrometers across, much less than a millimeter. We have a relatively thick barrier separating the ocean waters, and I'm thinking here we're deep inside the vent. The ocean waters percolate in, the hydrothermal fluids percolate in. We have a barrier, and on one side it's relatively acidic, and on the other side it's fairly alkaline. And this is roughly what cells look like. So cells are pumping protons out all the time. And so on the outside, they're relatively acidic, and on the inside, they're relatively alkaline. And that difference across the barrier is about 3 pH units, give or take, in both cases. And in both cases, it's acid on the outside. So it's very analogous. It's inspiringly analogous, but there's no reason at all to think that it should be any more than analogous. The question is, could it be? Is there an experiment that we can do to test whether this might work? Well, there are some very simple experiments that we've started doing in the lab. And we can produce these hydrothermal structures containing iron sulfur minerals. And the question is, well, can we use that structure and the difference in pH across the barrier to drive the formation of organic matter? It's very early days still, but I'll just show this. This is formaldehyde, which is right at the top of that energy barrier that I showed you. And this is within 2 hours or so. So we have a lot of controls we need to do, and I'm not going to talk about this work tonight, but it does look as if that structure might promote this reaction between hydrogen and CO2, and we will be continuing with that. What I really want to draw your attention to about these vents now, though, is that they're formed by a chemical reaction between rocks, the kind of rocks that you find in the oceanic crust, so rich in iron and magnesium, and water. So water will percolate down into the crust to depths of about 5 or 6 kilometers. So this is the ocean above. And they react with the rock, and it becomes metamorphosed. And it's converted from olivine, for example, into serpentinite. So this reaction is called serpentinization. So now we have warm alkaline fluids, and they bubble back up because they're buoyant. And when they get in contact with the ocean waters, they precipitate these amazing, they can be 60 meters tall, vent systems. So that's what's going on, and the only requirements really are rock and water. And so you would expect to see these kind of vents on any wet rocky planet. And there's even some evidence from our own solar system. So this is Enceladus, and on Enceladus you see these geysers periodically, and from the ions dissolved in those, you can work out roughly what the pH is, and the pH seems to be 11. And there are very few processes that are going to give a pH to an ocean which is 11, apart from this same process of serpentinization. On Mars, there's perhaps traces of methane, and that methane as well, it's either coming from life, which is difficult to believe, or it's coming from this geological process, reacting traces of water down in the rocks with the rocks themselves. And of course, Europa is another place to look, and again, it has massive oceans, and there are some signs that serpentinization is going on. So the real key point from all of this first half of the talk is that wet rocky planets will form this type of vent, bubbling with hydrogen gas. It should happen on potentially tens of billions of exoplanets across the Milky Way alone. These vents should have natural proton gradients, and those natural proton gradients should drive this difficult reaction between hydrogen and CO2. And so it begins to make sense of this broad shape. Why is it that these simplest cells are using proton gradients? Why are they? Why did it spread to everything else? This is a way of seeing it that makes some kind of sense. And again, it might not be true, but at least we can try to test it. But if it is true, then life elsewhere should face rather similar constraints. And so we can get at this question, why was it that the bacteria have been flatlining for 4 billion years? Why did life get stuck in a rut? And would it be the same reason elsewhere? Could it be something to do with the requirement for proton gradients? So it's what I like to think that John Maynard Smith would have called an evolutionary scandal. John Maynard Smith was a great evolutionary biologist, one of the doyens of evolutionary biology of the 20th century, and was at UCL as well, where I am, and so I am proud of that. He worked largely on the evolution of sex, which was very much an evolutionary scandal, but I think he saw this problem in similar terms as well. The problem is that all complex life is composed of this one cell type, the eukaryotic cell, which arose only once in the entire 4 billion years of evolution. Now, maybe they arose at other times, but we've not seen any sign of them. We can't, nobody's ever found one. So, to the best that we know, they arose once. And all eukaryotes share all kinds of universal traits. We're all sexual, for example. And bacteria don't really seem to evolve any of those traits at the morphological level in the same way. So the scandal then is that if all of these traits evolve step by step by standard natural selection, and each small step offers an advantage in the conventional way, then why don't we see any of them arising in bacteria for similar reasons? It's not obvious. Now, I'm not criticizing natural selection at all. The question is, what are the constraints that are operating on natural selection, and is energy one of them? Just to give you a sense of the problem here, eyes, for example, they're often dismissed by creationists as being, what use is half an eye? And you have the feeling that natural selection is laughing at them because these are all different types of eye. This is, for example, this is an eyeless shrimp. It doesn't have eyes at all, but it's got these photosensitive strips on its back. This is Euglena. It's pond scum. It's a single-celled alga. This is its eye spot. This is another remarkable, this is a single-celled protist, and this is the retina here. This is all inside a single cell. This retina is made of chloroplasts that are usually used for photosynthesis, but it's cobbled together a retina from them. And the cornea here is made of mitochondria, the powerhouses, but they're used as they're used as part of the cornea for this eye. And there's all kinds of other different types of eye. These are scallops here, for example. They're astonishing things. In animals, they can trace a common ancestor, which is a light-sensitive spot on some kind of an early worm. And there are some regulatory genes in common, but those regulatory genes independently recruited all the rest of the genes to evolve a complex morphological eye independently. So the eye of an octopus here is very similar to our own eye in its overall structure, but it is completely originally evolved. It doesn't share a common ancestor. This is convergent evolution. So what was going on then down here? Why was it that all of these, in bacteria and archaea over 4 billion years, with all of this genetic variation, why wasn't it that they could come up with something like half an eye? Why can't they come up with half of phagocytosis or of a nucleus, or all kinds of traits that complex cells do and that they don't do? So what was happening down here that wasn't happening over there? Now, I like to think of this as the black hole at the heart of biology. It's not just at the level of multicellular organisms, it's also at the level of the cells themselves. So this is planktomycete. This is a relatively complex bacterium. It even has a structure that you might just be able to make out. It's compartmentalized. It's being called a little bit like a nucleus. It's not really very similar to a nucleus, but it's got a relatively complex structure. But it's roughly to scale, which is why you can't see it very well. And this is Euglena, the pond scum I mentioned a moment ago. You don't need to know what any of this is to appreciate that it's on a completely different scale to the bacteria, with an awful lot of things going on. These are the chloroplasts, in fact, and this is the nucleus here. But we don't need to know what any of that is to appreciate that there's a big void in complexity, even at the level of single cells. So what was going on? The other thing I want to call your attention to is within the eukaryotes themselves, the level of complexity is really strikingly similar in very different types of cell. So this is paramecium here, which is, again, you'll find it in local ponds and things, and this is a pancreatic acinar cell. And they're roughly the same size, and at a quick look, they look roughly similar in complexity. I often ask my students in the first year when they start, how many genes do they think that paramecium has? And I've been shocked at the answers I get sometimes. They range from 10 through to a few thousand. The actual answer is 40,000. They have 40,000 genes. That's twice as many as we have. So the level of complexity in terms of protein-coding genes in single-celled pond scum is quite striking. Now, I'm making fun of the students, but I should make fun of the professors as well. When the Human Genome Project was just coming to fruition in around about 1999, 2000, there was a sweepstake to try and guess the number of genes that there would be. And the average number that people guessed was 80 to 100,000. And of course, now we know the answer was 20,000. And I have to say, on behalf of evolutionary biologists, that the evolutionary biologists in the late 1960s and 1970s, on the basis of mutation rates, had demonstrated that the human genome could not have more than 30,000 genes. unfortunately, the scientists who are working on unraveling the genome itself didn't know that literature. That's another point about science. It's very difficult to keep abreast of a very wide literature and have an idea, if you're in one field, of what people have been doing in another field. It's very, very difficult to do that. Another thing that we all have in common, all eukaryotes are sexual. Sexual in the sense that cells fuse together and the nucleus fuses together, and we don't see that at all in bacteria. Now, there are quite a few eukaryotic cells that appeared not to be sexual. So this is amoeba, for example, and until 2011, so this is Dan Lahr here. I imagine that he's out looking for amoeba in the woods, but we knew they had some genes that were linked with sex, but they'd never been caught in flagrante, and Dan finally caught them. Here they are, amoeba having sex. I have to say I was a bit disappointed. I don't know quite what I imagined, but I kind of thought they'd be full of bristling pseudopodia or something, an excited amoeba, but that's not quite how it looks. Anyway, the key point I have to get to the argument here is that all eukaryotic cells have or had mitochondria, and so this link with energy can explain why it is that all complex cells only arose once. So all of these cells do not have mitochondria, they lost them. And it's another illustration of science. There was a very good hypothesis from 30 years ago saying that perhaps these were primitive and they never had them. And testing that hypothesis showed that it was wrong. And so the importance of being wrong in science is vital as well, because we know an awful lot more about these cells. And it turns out that they did have mitochondria once upon a time. Now, Lynn Margulis showed really in 1967, 50 years ago this year, that mitochondria derived from bacteria. Now, this is Lynn Margulis. She married Carl Sagan. This is a kind of anti-celebrity marriage, you might say, because neither of them were famous at the time. Carl Sagan, I'm sure everybody knows Carl Sagan, but Lynn Margulis was a very celebrated biologist, evolutionary biologist, celebrated and worried about, because some of her ideas, the ideas to do with mitochondria and chloroplasts being derived from bacteria, were true. Other of her ideas turned out not to be true. It's another theme from science, though, that she persisted with these ideas, and the ones that were true, after 20 or 30 years, everybody accepted them to be true. But it required a real dedication on the part of an individual who was being really tormented. There were papers published which dismissed what she was saying, and it turned out that she was right about a lot of these important factors. She was asked towards the end of her life, she died a few years ago of a stroke, she was asked, don't you get tired of being called controversial all the time? And she responded to say, well, I'm not controversial, I'm just right. So this was the derivation of the mitochondria. They came from bacteria. And the host cell that acquired them in fact, just 2 months ago, less than that, a month ago, we now know the host cell was an archaeon, related to the Loki archaeota. So this is Loki's castle off the coast of Norway. And about a year or so ago, there was a paper showing that the host cell was very closely related to the Loki archaeota, which were dredged up from down there. We've never seen these cells. All we have is a genome sequence. This is what's called environmental metagenome reconstruction. And recently, more systematically across the world, a couple of weeks ago, they've come up with another paper showing that this is now a superphylum of all kinds of different groups. So the Loki archaeota are here, the eukaryotes are here, we're very closely related. We now have also the Thor archaeota and the Odin archaeota and the Heimdall archaeota, so we've got a thing going here. So this is how the tree of life now looks, and this comes from a good friend of mine, Bill Martin, one of the most brilliant scientists, I think, of his generation, and not always fully appreciated because he can be a bit prickly. But this was his conception in 1998, that we have separate origins from a vent, from a hydrothermal vent, from the kind of vent system I've been talking about, independent origins from there, from a common ancestor within that vent, of the bacteria and the archaea. And then the eukaryotes are a singular, this is a single event here, an endosymbiosis in which a bacterium gets inside an archaeon. And that gives rise to all of this complexity within the eukaryote. So it's a single moment, really, in evolution. And it started something like this. So this is the only example that we know of bacteria living inside a bacterial cell. So this is cyanobacteria with some thylakoid membranes. The key point about it is, the reason it matters is that you have a population of cells in there. And when you have a population of cells, well, what bacteria do, if they're just growing, imagine this is a population of bacteria, and this yellow cell here, it's got a mutation. It's wiped out a gene that it doesn't need anymore, and so it's got a slightly smaller genome, and it grows slightly faster. And so over time, it comes to dominate. And by here, pretty much all the cells in that population have lost that gene because they don't need it right now. But then the conditions change, and they do need it again, and so they pick it up from somewhere in the environment by a process called lateral gene transfer, and before you know it, you're back where you were. But what happens here, if you've got the same population of bacteria inside a cell, they lose that gene, and they never need it again. And so they just waste away, getting smaller and smaller. It's a genomic streamlining going on. And we know of plenty of examples of this kind of thing. Typhus is a very good example of it. So this is Napoleon's retreat from Moscow, where the army was almost obliterated by typhus. Now, typhus is caused by a bacterium, an intracellular bacterium, rickettsia. This is rickettsia invading a cell here. And the key point about rickettsia is that it's got a tiny genome. It's about a fifth of the size of most bacterial genomes. It's just 1 megabase in size. And that turns out to be general. So this is the range of genome sizes of free-living bacteria. Actually, it comes across to here now at about 12 megabases. And here are these intracellular ones, the parasites and the obligate symbionts, generally less than 1 megabase. So most of this work's been done by Ryan Gregory, but you see a very striking difference that if you're living inside another cell, you throw away everything you don't need and you become very small and very dedicated to a particular way of life. And that's what happened to the mitochondria. The mitochondria started out as bacteria and they ended up with a genome of their own. A very, very tiny genome. 99% of their genes have gone. And it's a really interesting question. Why did they retain that 1%? And the answer it comes from John Allen, who's here in the audience. And this is another wonderful idea that John put forward first in 1995, and it's still not generally accepted, though it should be. I don't have time to go into details, but effectively all of these mitochondria here have their own genes, which they use for controlling respiration locally. So the membrane potential on these, the actual electrical charge across those, is about 150 millivolts. And if you were to shrink yourself down to the size of a molecule, then that would be equivalent to being a field strength of 30 million volts per meter, which is equivalent to a bolt of lightning. So the charge on these membranes is enormous, and they seem to need genes to deal with that kind of charge. And so they've kept them. So just for the last couple of minutes, what's actually going on then? This is the situation. If we see giant bacteria, they have thousands upon thousands of copies of their complete genome. It's a thing called extreme polyploidy. They seem to need to have genes next to that membrane to control respiration in the same way that eukaryotes do. But eukaryotes, this is a eukaryotic cell, this is the nucleus in blue here, the red are the mitochondria, and the green dots are the mitochondrial DNA. So they have these tiny mitochondrial genomes supporting a massive nuclear genome. So what's happened is we have a genomic asymmetry where all these genomes are the same size and shape in bacteria. You expand up, and they just copy and copy and copy, and there's no evolution going on. They all remain the same. But here, over time, these genomes have become tiny, and they can support, then, energetically, that massive nuclear genome. And so why did complex life only arise once? Well, it required, at the very beginning, a singular endosymbiosis, where one cell gets inside another cell. And this is improbable. We know of this one example. And then after that, well, you know, there are no known surviving intermediates of this process. They've got to find a way of syncing up their lifestyles and living together. It reminds me of Sartre's comment that hell is other people. That's what was going on here. And this is equivalent to that last common ancestor. It had everything. So this is the black hole at the heart of biology. And this is what is very difficult to get at how it happened. But I think another important take-home message is that when we're trying to understand what's going wrong in diseases and things, it's important to know how all of the parts of a cell relate to each other. How do they work? How do they function? How do they evolve to be that way? And if we can't understand that, it's difficult to understand how they go wrong. And at the moment, we don't really have a good idea of exactly how they all evolved in the first place. As I said earlier, we have ideas, but we don't know which is the right answer. So I've said that life arose only once, complex life, and it was very rare. And that may depress some of you. And so I want to cut you a little slack, because this was discovered in 2011, and it's not clear at all what it is, which is another lovely thing about science, is we don't know what it is. The Loci archaeota, we have a genome, but we don't know what the cell looks like. We've never seen the cell, and so we try and piece it together from the genes. In this case, we know what the cell looks like. It's this, but we don't have a genome, so we don't really know what it is. Now, this is not a normal cell. It's got what looks like a nucleus here, quite large. It's got some things that look a bit like hydrogenosomes, but they could be intracellular bacteria or something. It's got membranes, it's got a cell wall. It looks like a fungus of some sort, probably a highly derived fungus. But when you look more closely, you realize that the nuclear membrane, well, it's a single membrane. It should be a double membrane, but it's just a single membrane, so that's odd. All of this dappling here, these are ribosomes, which are the protein building factories in shells. They're never found in the nucleus, and yet here they're all in the nucleus and outside the nucleus too. That's odd too. This nuclear membrane actually fuses with the cell membrane, and there's a lot of things wrong with this cell, and it might be that it's all an artifact of electron microscopy preparation, we don't know. Or it might be that it's somehow recapitulating, that we've got cells within a cell and that it's become larger. These are normal-sized bacteria here. It's become large. It's developing a nucleus. It's kind of recapitulating eukaryotic evolution. The downside is they've been trawling this trench off the coast of Japan for the last 15 years, and they only found one like that, and they've never found another one. So it's very rare, and perhaps complex life has arisen on multiple occasions, but always went extinct before it got very far. So I want to finish with this. So I'm sure some of you will recognize this as the pale blue dot, which is there. That's the Earth. And this was Voyager 1 in 1990, when it was beginning to leave the solar system, and Carl Sagan managed to persuade them to turn the camera around to face the Earth for one last time and take a picture of the Earth. So these bands are actually light scattering artifacts, and they're not true at all. But that's the Earth. And Carl Sagan, with his characteristic eloquence, said that everything which we know about, everybody we've ever loved, everybody we've cared for, everybody who ever lived, all the civilizations and the empires and the tyrants and the religions and everything that's happened on this planet from the very beginning happened on that pale blue dot. The thing that strikes me about it as well, though, is how inscrutable it is. How could we know anything really about that? And science itself the world is inscrutable. It's almost impossible, it seems, to know anything about all of this, and yet we do. And we know about the world, and we know about atoms, and we know about subatomic particles and quarks and membranes and strings, and we know at the other side about black holes, and we know about the structure of cells and the proteins that make them up and how they work. And this is from the process of science over hundreds of years, finding ways of asking probing questions. And it works a little bit like natural selection, in the sense that there are always competing hypotheses, competing ways of seeing a question. And it's the testing of those questions, just as natural selection distinguishes between different versions, some of which work better than others. In science, there's a ratchet-like way of improving our knowledge over time and beginning to get at some answers. And so a lot of science is now, in effect, in dusty textbooks, because it was established a long time ago. But where science is really at is in this process of discovery, in the process of trying to understand new things about the world. And that process is inevitably about competing hypotheses and arguments and different ways of seeing the world. And that's how it needs to be. But it's also a dangerous way of presentation, I think, in the modern world, where experts are dismissed as being meaningless. We have to find a way, I think, as a society, of somehow getting people to think more clearly about what science is as a way of thinking about the world, a way of questioning the world, a way of getting at inscrutable information. And this is one of the reasons why I think books are valuable, why any form of public engagement is valuable. It's vital, I think, if we want to keep the whole Western way of life alive, which is based on this scientific understanding of the world. We have to find a way of getting everybody to appreciate the process that's going on. And when they see two scientists who are arguing with each other, appreciate that they're arguing about details and they're arguing about how to move forward. They're not arguing necessarily about what we know from the past. And I think that's a key message to end with. And thank you all very much.
Thank you very much, Nick. Terrific.
Afraid I've overrun slightly. I hope we still have time for a few questions.
We do. So Nick has very kindly agreed to take 10 minutes or so of questions. Could I ask you, please, to use the microphones if you want to ask a question, and if you can, please stand up. So who wants to ask a question? We have one down here on the left. Okay.
Hello, thank you very much. I just wondered about the chloroplasts. I always understood that chloroplasts were another sort of endosymbiont, an extra one. I wonder if you could sort of clarify that for me, please.
Yes, they are an endosymbiont. They derive from cyanobacteria, but it was not a singular event that gave rise to complexity. So the acquisition of mitochondria gave rise to all eukaryotic cells, every one of them. And then in one group later on, they acquired a cyanobacterium which went on to become the chloroplast. So yes, it's a separate endosymbiosis, but it didn't affect the overall kind of direction, if you like. It affected everything tremendously in the sense that it allowed photosynthesis to take off in a completely different way, and so it changed the way the world went. But in a conceptual sense, it doesn't alter the fact that it was a singular endosymbiosis at the origin of the eukaryotic cell. That came later.
Question here.
I wanted to ask you whether you're aware of anybody who's actually mixed together archaea and bacteria under a variety of conditions to see if you can actually observe a bacterium getting into an archaea. Has anyone attempted that, or is it regarded as so improbable?
Well, I would love to try and do it. It's not the kind of experiment that's easy to get funded, and you can't just inject one inside another. I mean, you could do that, but it would fail because you need to have a way of making a living inside another cell. And so the ideas are that there would be lots of gene transfers going on, and you would need to set that up in the lab. I have met several people who've told me that they're doing exactly that. And the strange thing is, I've never seen any of them since.
Is it fair to conclude that because it took so long for eukaryotic cells to arise, that if there is life elsewhere in the universe, it's far more likely to be prokaryotic than eukaryotic?
I would imagine
and therefore we don't need to worry about E.T.
Yes, or Vogon poetry readings, yes.
There's a question over there.
I was wondering if some cells have developed in a way that they don't need mitochondria anymore. Is it possible for cells to evolve in a way that they take this shortcut from the beginning?
Well, I would say no, it's not possible. And that's a hypothesis that may well turn out to be wrong if someone shows a complex cell tomorrow that doesn't have and never had mitochondria. So we know of a lot of cells that don't have mitochondria. So it's perfectly possible to be complex and not have mitochondria. The question is really, is it possible to become complex in the first place without having mitochondria? And the energy difference that having mitochondria makes is actually immense. I didn't really mention it. It's about 100,000-fold difference in the energy availability per gene that eukaryotic cells have above bacteria. So it allows you to just ramp up on a massive scale and to have far more protein synthesis going on, to have far more genes, to be able to have lots of bad genes probably, and it doesn't affect you very much because you've just got bucket loads of energy sloshing around and you can deal with it. So once you've got all of that, and most of these cells that have lost their mitochondria are phagocytes. And that's important because once you're a phagocyte, you can go around and engulf other cells. which means you may not get much energy out of eating them, but you can just eat more of them and you'll be all right. Whereas if you try to do that as a bacterium, well, there are no bacterial phagocytes. The whole process of becoming a phagocyte is so energetically costly that what I'm suggesting is that's the barrier, really, that you can't be. And so the cells, the bacterial cells that just ferment, they exist as well, they're tiny because they can hardly extract anything. They can't eat other cells. They just have to use the resources of the environment. And so they have the smallest genomes of any known bacterial cells.
Okay. One last question here in the middle.
Thank you. Life's been around for 4 billion years, but eukaryotic cells have only been around for about 2 billion years.
Yeah.
But that fusion event took place not long after the Great Oxidation event. So therefore, once you had oxygen in the atmosphere, that sort of fusion event is possibly more likely.
Well, that's what Lynn Margulis thought originally. That
The
was her
origin
original
of a
hypothesis in
eukaryotic
1967.
cell could be a little bit more, likely from what you're saying.
Well, that sounds reasonable on the face of it. The problem with it is we don't see multiple. That's exactly what you would expect to see. Once you have oxygen, you would expect to see, for example, cyanobacteria might evolve directly into plants because they're already photosynthetic. They're already halfway there. You would say they were pre-adapted to that lifestyle. Why can't they just go on? Why don't they pick up another cell if they need another cell, if it's much easier to do that with oxygen? What about fungi? Why can't they derive directly from osmotrophic bacteria that have a similar lifestyle? Why don't animals arise from phagocytic bacteria or some kind of predatory bacteria? What actually that theory predicts is that we should have multiple origins. After an environmental bottleneck like oxygen levels rising, we should then see multiple origins, maybe not tens or hundreds of them, but certainly we should see 2, 3 or 4 of separate origins of complex life, but that's not what we do see. And it's not at all clear exactly when the eukaryotes arose or whether it was linked to that period right after the Great Oxidation event. And also, over the last 10 years, we see oxidation then, but not much oxygen. Most of that oxygen gets consumed by oxidizing the rocks themselves, and that's why we see these red beds and so on. But in the oceans, there's very little oxygen for another billion years, really until the time of the Cambrian explosion when we see animals very abruptly, and that probably was linked to oxygen.
Okay. I think we better stop the questions there. We now come to the formal presentation. So Nick, it's my pleasure to present to you the scroll and also the medal.
My word, thank you.
And finally, the check. Thank you. Wonderful. Congratulations. Wonderful. Terrific.
Thank you.
So, ladies and gentlemen, a private function is due to take place in this lecture hall shortly. In order for our staff to prepare the hall, we would be very grateful if you could make your way to the exit as quickly as possible. Unfortunately, Dr. Lane will not be able to remain to answer any further questions. Guests attending the private function should make their way to the City of London 2 room, which will be on your left as you exit there. Thank you very much for coming, and I hope you have enjoyed the evening. Thank you.